So, one of the things that both of you sort of nicely encapsulated is a lot of the work that we're doing on the drug substance side, in particular with the new polymerases, the capping reagents, they're really helping us to get off on solid footing with our upstream processes, right? To help kind of eliminate some of the extra purification hurdles we might face, right, on the downstream. But I guess my question for you is sort of also thinking about some of the quality understanding, right, and the scientific understanding that we have today of these novel approaches, right? And Khaled, you beautifully summarized the fact that risk is a consideration here as well, as we're trying to explore novel materials, right? We know T7 polymerase, we know kind of how it works, right? We know its pitfalls. But as we start to advance into different polymerases, what do we know, right? What don't we know yet? So maybe, Francoise, I'd love to just sort of get a general sense from you of how well, right, do you think we as the industry, as well as our suppliers today, really understand the quality and or the performance, right, of some of these novel raw materials today? And how should we, what best practices should we be enacting to sort of improve our knowledge and understanding of these products?
So first of all, what we have done maybe now 2 years ago, it's really to move outside of animal origin, antibiotic content. So that was really the first step, to be sure that we were getting raw material with proper quality. So animal origin free, antibiotic free, and also with the proper grade, GMP grade, because that was not the case at the beginning. And also, for sure, all the knowledge about the scale and the price, for sure. But what we are facing today is really to better understand, as Khaled said already, the way the raw material is working, in the product, and the way, we can also better understand between in vitro and in vivo model, how we can do the translation and how we can predict better. Because it's not only to get 100 of lipids if we can't be sure that at the end it will work well. So it's how we understand the model, and also how we can build new model, and this is what we are doing with Sanofi now, how we can have model to better accelerate and predict as soon as possible, the behavior of the material to ensure that we are moving on the right direction, and also to avoid huge investment on this material if it's not...
right, if it's not going to do what you need it to do. So
two sides for me: better understand the way the material is manufactured with the CDMO, and also how the material is used in manufacturing, and how the product, the behavior of the product at the end.
Absolutely. Khaled, I want to ask you a similar question, but I know that you've also done a lot of work, right, with USP and kind of homing in on the work that the growth in the polymerase market, right? And looking at the quality of polymerase, even just T7 polymerase, right? Overall. And one of the core questions that I have is thinking about, how well do we understand the materials that we have today? How well do we understand the newer materials? And a huge part of that is understanding the quality profiles of them, understanding what we're met with from our suppliers, right? And how well our suppliers understand what they're supplying to us. So, you've been involved in providing guidance on the quality of mRNA and the T7 polymerases. Can you kind of give us insights on how these polymerases are differing, right, in terms of quality, and what have been some of the biggest learnings you've taken away so far? Kind of a loaded question.
It depends. That's fine.
Yeah.
It depends who you ask. If you ask the manufacturers of polymerase, they're going to tell you we have the best thing, the best quality. If you talk to the users, I don't know what I'm getting. So there's this premise that all T7s are alike. They are not. So if there's a couple of things. One is if you take all the CFAs from different vendors of T7, each one is completely different. They are not aligned, and there's no synergy between all of them. So when you are starting a raw material program, you really have to build it from inside as the innovator or the CDMO to receive it. You really cannot rely on the supplier, because Otherwise, you're going to corner yourself to one supplier, because when you go to another supplier to have a backup, their test is different, their activity is different. There's really no. So most of companies, what they have done is they actually do a functional testing where they do a small IVT reaction to make sure that I got X and I got Y, and that's it.
Yeah.
And part of it is it's very hard to measure the true activity of the enzyme, because if you look at the IVT reaction, there is about 3 different steps the enzyme change its activity. So it's really hard to measure each step, what you get. So they're looking at it overall. So that is an issue that the vendors have not harmonized how, what to report in terms of testing.
Okay.
And but also I'll take it one step. That could apply the same reasoning or the same thing that you've seen, because I've done that also for NTPs and capping agents. It's completely different. So you really, it makes it hard to have multi vendors to ensure that you have the supply chain. So one of the things that I would like to see, and I kind of propose, like, for example, for the NTPs, and I'll come back to the polymerases, whether it's a company's consortium or we ask the USP to do something, is to have, it's not going to be a monograph, because the monographs usually apply to things that are going to go into your body. So like sodium chloride, there is a monograph that you test it to a certain level and it'll be a USP grade, but that goes into your formulation to be applied into the body. So those kind of things does not apply, but we need something to standardize, to standardize the testing, to call it whatever grade we want to call it, because a GMP is a process, is not a grade. Testing a quality, you're testing it to a certain quality, so that is a grade.
Yeah.
And we really need to differentiate that. But back to the polymerases, that's one, with collaboration with Primrose, I was able to do the study where we took several T7 from several vendors and applied it under their conditions, same template, same reagents.
Yeah.
You get completely different results in terms of the quality of the mRNA. So,
Interesting.
people have to really understand that they're not all functioning the same. And so what I advise is to really adapt the QBD system to establish, and following ICH part 11, ICH 7,
Yeah.
to start your own internal raw material program and link all of the data that you get to your process and to your product. So it's I mean, I spoke about that multiple times, and I do have a system where it builds up straight from the quality attributes all the way down to the critical raw material attributes, and how do those impact your process, and does your analytical methods that you use for the incoming raw material actually support what you're saying, and are they fit for purpose for that?